Hello everyone, I'm Khajeh. I'm a first-year PhD student at the UIC Department of Bioengineering. In this video, I'm going to present our research with the title of Is Gut Metabolome More Informative than Microbiome in Predicting Host Phenotype? This is a joint work with Derek Ryman and my advisor, Professor Yang Dai. Human microbiome is the aggregation of all microbes living on the surface or inside of the human body. These microbes reside in different areas, including the mouth, lungs, stomach, intestines, sex organs, and skin. In fact, these microorganisms are crucial to human health, and composition change in human microbial taxa is linked to various diseases, such as inflammatory bowel disease. The focus of this study is specifically on IBD prediction. Machine learning approaches have been developed using microbiome data to predict phenotype. However, the microbiome is noisy, which results in low prediction accuracy. Existing works have suggested that functional profile of microbes are more suitable for exploring host microbiome interactions and host phenotype prediction. Recent experimental technologies have made the measurement of metabolome possible. That is what we are going to use to improve the prediction accuracy in our model. The metabolome is the complete set of small molecule chemicals found within a biological sample. Metabolites are the final downstream products of genomics, transcriptomics, and proteomics. The metabolome has been shown to be more predictive of host phenotype than the microbiome. However, metabolome is a high-dimensional profile, which makes it challenging to train robust machine learning models. In this study, we are going to use autoencoder to derive low-dimensional representations. We use autoencoders because the standard dimension reduction method, such as PCA, is a linear transformation of data, and therefore it may not sufficiently capture the information. On the other hand, the autoencoder is a neural network capable of representing complex relationships among different features in the data. This figure is an overview of our proposed methodology. As mentioned, the microbiome and metabolome will be extracted from each biological sample, and their combination will give us the combined profile. Each of the three data types, microbiome, metabolome, and the combined profile, can be used as an input of the machine learning algorithm. Our machine learning algorithm consists of an autoencoder for dimension reduction and a multi-layer perceptron neural network for phenotype prediction. The dataset used in this research is from another study of concurrent profiling of the microbiome and metabolome in two IBD cohorts. One cohort is used as the train set with 121 case samples and 34 control samples. The other is used as the external test with 43 case and 20 control samples. This table shows the statistics of the data. The number of microbiome features is 201 and the number of features for the metabolome is 8,848. The biological samples are divided into three categories: ulcerative colitis, Crohn's disease, and non-IBD. We treat both ulcerative colitis and Crohn's disease as a single category. Therefore, we have a binary class prediction. Three types of autoencoder are designed for the microbiome, metabolome, and the combined profile. The three autoencoders all have two hidden layers and a latent layer. The hyperparameters of autoencoder were tuned using tenfold cross-validation. For microbiome and metabolome profiles, standard autoencoders were used with different cost functions and activation functions. Since the microbiome data are represented as relative abundances of operational taxonomic units, we used softmax activation function for output layer. Therefore, the sum of the total output from the autoencoder is 1. In addition, KL divergence is used as its cost function. As a metabolomic profile consists of metabolite feature abundance values, the metabolome autoencoder used the mean square error cost function and linear activation function for the output layer. For the combined profiles of microbiome and metabolome, a multitask autoencoder was designed to encode and decode each data type simultaneously. Technical details of autoencoders are represented in this table. Hyperparameters of multilayer perceptron neural network are selected using the grid search method after a 20 cycle of 5-fold cross-validation. Training is done using the learning rate of 0.001 and the adaptive moment estimation optimization algorithm on the training data. Evaluation is done on the external test. To visualize the effect of the autoencoder on each dataset, PCA plots with the first and the second principal component is prepared before and after applying autoencoder to each data type. Here, the yellow dots represent non-IBD samples, while the purple dots indicate IBD samples. Clearly, after the dimension reduction step, important information has been retained in both data types. Furthermore, the yellow dots are more tightly clustered after using the autoencoder. To evaluate the performance of models trained on the autoencoder-derived lower dimensional profiles, we considered 3 scenarios for our research experiment. First, raw features from the microbiome, metabolome, and the combined profile were used as the input into the multilayer perceptron neural network. So we developed 3 different models. The phenotype prediction accuracy of the developed models is represented in this box plot. The x-axis represents models. The y-axis is the AUC value. Boxes in green, blue, and yellow represent the combined profile, metabolome, and microbiome respectively. As illustrated, the model based on the combined profile achieved the highest accuracy. Also, the model developed using metabolome is more accurate than microbiome. Our proposed method is the second experiment. The values from the latent layer of the autoencoder will be fed into the multilayer perceptron neural network for each data type. As before, the model developed based on the combined profile achieved the highest accuracy. Moreover, the model based on the metabolome achieved higher accuracy than microbiome. The third scenario is used Mimonet framework developed in our lab. Mimonet is a neural network model developed for deriving the relationship between microbes and metabolites for predicting metabolome from the microbiome. First, we prepared module profiles derived from Mimonet. These modules can be considered as a lower dimensional representation of the data. Then these modules used as the input of multilayer perceptron neural network. Again, the model developed based on the combined profile achieved the highest accuracy, and the model developed using metabolome is more accurate than the one developed using the microbiome. Here, results from all experiments can be seen in a single plot. The x-axis represents 3 different models. The y-axis displays the AUC as the phenotype prediction accuracy metric. As can be seen from the results, our proposed method based on autoencoder resulted in the highest accuracy. In conclusion, in this research, we showed that combined profiles and metabolome have higher predictive power than microbiome for IBD. Also, autoencoder is a practical solution to effectively overcome the problem of high dimension in metabolome data. Additionally, deep learning models are promising tools to integrate microbiome and metabolome data types. And finally, models developed using autoencoder are more robust. For the future, we can evaluate our model using other datasets. Thank you for your attention.